All right, guys. So, which of the following best describes the hydrolysis of carbohydrates? Till this day, some people don't know what the word hydrolysis means. Are you one of those people?
No.
No. What does hydrolysis mean?
When you add
Add water so you break up on
bond.
You break it down by adding water. What's the opposite of hydrolysis reaction?
condensation.
Yes, or dehydration synthesis. So look, hydrolysis means you have to add water. So it's not A, it's not B. It is C. Which of the following best describes a characteristic of DNA that makes it useful as a hereditary material? Well, of course, DNA is special, right? It has what we call a double helix. So that means we're going to have complementary base pairs, which is very important. Look for something along those lines. D. Very good. Okay. Do we see a big difference in the number of the affected males and females here?
No.
No. 3 females,
2 males.
2 males. It's not a huge difference. Okay. Are we skipping generations?
No.
Yes, we are. Because 5 and 6 are not sick, but their kid is sick. Right? So here we are skipping generations. And if we skip generations, we call that recessive. Okay, so we're looking right now for recessive. And we just said that gender is not related. Now, I'm going to go with B, but I must also double check one more time. Because the cross between individuals I1 and I2 produced an affected offspring. Yes. This is called skipping of generations. The parents are not sick, yet they have a sick kid. That's the indication that the disease or the disorder is inherited in a recessive manner. Okay. To investigate bacterial metabolism, a researcher divided a population of bacteria into 2 cultures, and both cultures have glucose, which is food for the process of cellular respiration. He put a chemical to one of the tubes and he measured the pH at 5-minute intervals. Okay. If you are asked to graph this in the FRQ, what type of graph would you use? A line graph would be best. Because you have a single treatment that's been monitored over a lot of time, from 0 to 40 minutes. Okay. Now, the bacteria broke the glucose down into lactic acid. You know what that means? Anaerobic respiration. Now, lactic acid does what to the pH? It will decrease it. If you have more lactic acid, you will get less pH. Which of the following best describes the process by which the bacteria are breaking down the glucose to produce lactic acid? It is going to be fermentation, right? Does fermentation require oxygen? No oxygen. So, the answer is A. How come it is, can it be C? Do bacteria have mitochondria? No. No, they don't. And without mitochondria, can they do Krebs cycle?
No.
Of course not. And the H gradient is only there if we are talking about the inner mitochondrial membrane, and they don't have that. 5. Which of the following was the dependent variable? Dependent is the one that we are observing. We are observing it.
The pH.
Yes, we are. Are we controlling the pH? No. So it's dependent because we don't control it. We control the time. That's called independent. We control how much glucose to put. That's also called independent. Now, we don't control how much lactic acid is produced, but we are not observing for it. We are only observing the pH. Which of the following best represents the data in table 1? Like I told you, you must use a line graph if you see time in this much amount. Line graph is going to be best.
B.
So B. Exactly. Okay. Based on the data in table 1, which of the following is the earliest time point at which there is a statistical difference in the average pH between the control and the treatment groups? Now, I recommend you look at the figure that was already given. I think it's easier for you if you just use the figure. I believe. Let's check. Where are they not overlapping? Are they overlapping at 35? No. But they're overlapping everywhere else. Now, let's make sure with the table. At 35. You must do the plus and the minus. So you can get 7.63 and 7.67, 7.58 and 7.62. They're almost overlapping, but not overlapping. So we are right. At 35, they start to become significantly different from each other. Okay. 35. Okay. According to the data, which of the following best explains the results of the experiment? Do you guys understand the table? Over time, the pH drops. Okay? The pH drops because of what? Of more lactic acid. The pH of the treatment. Okay, so A is correct. The pH is dropping. Is the pH becoming higher؟ مرحبا علي. سلام. شلونك علي؟ تسلمين. شكرا. Is the pH becoming higher or lower?
No. It's A. Yeah, it's A.
How come it's not C?
Wait, it is C or not C?
No, he said, how come it's not?
Well, I trust your answer. It's either going to be A or C. Let's decipher which one and let's see why. Did the pH of the culture become lower? It did.
Yeah.
Did the chemical increase the bacterial metabolic rate?
No.
Well, it did. Look, they both have that. They both have the chemical increase the metabolic rate, right? That means this is a correct statement. But the question is, which one comes first? The pH becomes lower because, or the chemical increase because?
No, the pH because.
I think it's A is the one that makes more sense. The pH becomes lower as a result of. Let's make sure. Yeah, it is A. Okay. All right. So 9. A mutation in the gene coding for a single polypeptide enzyme results in the substitution of the amino acid serine, which is polar, with a non-polar one. When researchers test the catalysis, which means the enzymatic reaction of a normal enzyme and a mutant one, they find that the mutated enzyme has a lower activity. Makes sense, right? It's mutated, it has a different amino acid sequence. So why is that? Does the mass of the enzyme matter?
No.
It does not. We are changing the tertiary structure. We are. Which results in a different folding.
So it's B.
So it's B. Pitcher plants are carnivorous. That means that they eat animals. And they grow in the area where the soil contains low levels of key nutrients like nitrogen. So to get the nitrogen, they will capture prey and they will digest the prey inside. The traps of one species of the pitcher plant don't have a digestive fluid. And instead, what they do is they provide a place for the bats to sleep. The bats will, give them feces which have nutrients. They get the nitrogen from the feces. So the bats are getting helped from the pitcher plants, and the pitcher plants are getting helped from the bat. What do we call that? Mutualism, exactly, because they both benefit in the process. A particular genetic disorder is associated with a single gene with two alleles. Right now we're talking about Hardy-Weinberg, because Hardy-Weinberg always talks about two alleles. Individuals with two recessive alleles are affected. The prevalence of the disorder. What letter is referred to when discussing the prevalence of the disorder? If it's recessive, Q or Q squared? If it's disorder, Q squared.
Mr. Repeat the question.
Look, first of all, our disease is what? Recessive. So it's going to be either Q or Q squared. Q is for what? For the allele. Q squared is for what? For the disorder. So right now they gave us Q squared. So Q squared is equal to 1 over 6,000. So Q is equal to the square root of that. Okay? And the square root of that is going to be 0.0129. Now the question asks for the frequency. Now, of course, some of you might be happy and they see it's B and they choose it. Be careful. They're asking for the frequency of the carriers.
Wait, mister, why did you do 6,000?
Sorry. 6,600. Good point.
And it's going to be C.
And don't get too happy. Don't choose B yet. We're looking for the carriers. What are the carriers again? What's their genotype?
Recessive.
Nope.
Right?
2PQ. So 2 times P is what? 1 minus 0.0123. And a Q is 0.0123. Right? So do you guys know what this is? This is P. And that's Q. Good. I got 0.024. Which is C. Okay. What do you guys think the thickness of the arrow signifies?
ATP.
No. The thicker the arrow, the more stuff are happening, right? So as you guys can see, the arrow becomes thicker over time. That means that more molecules are involved, which is very typical of signal transduction pathway. That over time, more and more molecules get involved in the process. Now, blood vessels are surrounded by cells called smooth muscle cells. Nitric oxide is a gas that triggers a signaling cascade in the smooth muscle cells that makes the cells relax as the response. Which of the following is represented by the gradual increase in the thickness of the arrows? Okay, so if they become thicker, the molecules are increasing. So what do you guys think?
B.
Exactly. A student peeled the skins from grapes, exposing cells with membranes that are only permeable to water and small diffusible solutes. The student measured the mass of the peeled grapes. He then placed each peeled grape in one of 5 solutions. And after 24 hours, the student removed the peeled grapes from the solutions, measured their final mass, and also calculated the percent change in the mass. Question to all of you. Why do you think the percent change of mass when placed in distilled water was positive 13.48? Did the mass go in the grape or leave the grape? How? The grape became heavier by 13.48%.
So the solute.
So the water went in. How about the second one?
Out.
The water went out. The water went in. The water went in. The water went out. Okay. In the second experiment, the student placed a peeled grape into a solution containing both small diffusible solutes and solutes that don't diffuse through the membrane. Okay, so, right, inside the grape, we have less diffusible solutes. Outside the grape, we have more of them. But the osmolarity of the ones that don't diffuse is higher inside the grape. That means that the water is going to go this way. So the water is going to go toward the more osmolarity. So the grape is going to expand. Okay? The grape is going to expand. Look, these guys don't move. They just make the water move. Okay. Based on table 1, which of the following best explains the difference in water potential between certain solutions and the grapes? So, distilled water and NaCl, did they act the same way? No. NaCl and tap water, let's see. Did they act the same way?
No.
No. So we're going to cancel out A. Grape soda and NaCl.
Yes.
They acted the same way? Yeah, they both made the cell lose mass. So this is the cell, and this is the solution. And to lose mass, the water has to go to the solution. Why? Because the water always goes from high water potential to low water potential. Okay.
So it's B.
So it's B. So basically, what you have to know is that choose the least positive number and the least negative number. So the least positive number is the 2.8, right?
Yeah.
And the least negative number is how much?
-15.
Is -15 for the grape soda.
Okay.
Now I want you to basically add 2.8, right, to
-15.
-15 and divide them by 2. So like we're averaging them. How much do we get if we do that, you think?
5.5.
Can you just...
You add them or minus them?
You minus them because one of them is negative. The 15 is negative.
6.1.
That's negative 6.1%. So what number is the closest to the positive value? So what number is the closest to 6.1? That is going to be C.
Can I know why you took the least negative and the least positive number?
I have to be as close as possible to 0. So I have to be as close as possible to isotonic solution. Because in isotonic solution you would have a 0% chain.
How does the negative one make it 0%?
The negative 1?
Make it 0%. How?
It doesn't, but we are getting as close as possible to its own percentage. Look, if you get a 0%, which you don't, you have isotonic solution. So you want to choose percentage of mass that's as close as possible to 0%. So I took the least positive one and the least negative one. I add them, take the average, I got 6.1. Choose whatever is close to 6.1, which is 5.5. A student hypothesizes that the solute concentration of grape juice is higher than the solute concentration of the actual grape because the grape juice has added sugar. Based on the data in table 1, which of the following best evaluates the student's hypothesis? The solute concentration of the grape juice is higher than the actual grape. Let's see. Grape juice. Grape juice. Grape juice resulted in a percent change in mass that's positive. That means what? That this is the grape and this is the grape juice. That water went inside the grape. Why would water go in the grape? Because this one has higher water potential and this one here has a lower water potential. To have a higher water potential, you must have less concentration. So, is the student right or wrong? Here it says the solute concentration of the grape juice has to be higher. But there we just said what? Lower.
We don't support.
So we don't support the hypothesis.
Because it's the
Let's see why D is the case. Yeah, because the mass of the grape increased. That's exactly what the table is saying. Let's make sure. Yeah, okay. Good. All right, so assuming a negligible pressure potential, which of the following best predicts the net movement of the small diffusible solutes and water in the second experiment? They will move from higher concentration to lower concentration, right? That means they will do what? They will move inside the grape. They will move inside the grape. Good. Okay. How about D? How about D? Why cannot D be correct?
because
The water will also move inside the grape because the water is going to move to the area of higher concentration of non-diffusible solutes. Water always goes to the area of more... What the heck keeps texting like that? Sorry, guys. Okay. Yeah. Okay? You guys get it? Water moves in, and the solutes also move in. 17. Mercurial sulfhydryl.
Wait, it said it will move out or in? What will move out?
17. Mercurial sulfhydryl is an inhibitor of aquaporins. Do you guys know what aquaporins do?
Their water channels?
Yes. Which of the following is the most likely effect of adding mercurial sulfhydryl to a distilled water solution? Can the water still move? Not so easily, right? So, distilled water solution was causing the water to move into the grapes and making them expand. But now, without the aquaporins, can the water move as much?
No.
No. Okay. They will still get water, but not as much as before. Look, without aquaporins, water will still move, but will simply move slower. So D. Which of the following best explains why larger grapes have a different rate of water absorption? Whenever they talk about size and absorption, we're looking at the ratio of surface area to volume, essentially. So a larger grape has a low surface area to volume ratio. Okay? So what's the answer? Rate has to be slower, correct? It's B, you said?
Yeah.
Larger grapes must have lower ratio, not higher ratio. That means smaller grapes have a higher ratio. Which of the following best explains how some cells of an individual produce and secrete a specific enzyme, but other cells of the same individual do not? Look, all the cells have the same genes, but in some cells, that gene is active. In other cells, the same exact gene is not active. So it's not different genes. It's the same genes, okay? It's the same person. How can the cells evolve under different pressures, right? Let's see. Here what we see is that over time, the concentration of the product is increasing. But is the rate, which means the slope, becoming less or becoming more? Is it becoming less or more steep, the lines?
More.
How? If you find the slope from here to here, it's more than the slope from here to here.
So we start from 0.
So the rate slows down over time. So the rate will decrease. Does the enzyme get used up in a reaction? No, it does not. So it's not D. Okay. Pyruvate kinase, a key enzyme in the glycolysis pathway, is inhibited by the amino acid alanine. Okay. But the ability of the alanine to inhibit the enzyme is not affected by increasing the concentration of the substrate. It's not affected. Okay? You know what that means? Are we discussing competitive or non-competitive inhibition? Which one?
No.
Non-competitive. We are discussing non-competitive inhibition. Okay. Which of the following best explains the mechanism by which alanine inhibits pyruvate kinase? Yes, it has to be the allosteric site, right? It has to be. Good. Different species of bumblebee compete for flower nectar in their ecosystem. While the flowers vary in the length of the petals, bumblebees vary in the length of the mouth part called proboscis. Bumblebees demonstrate a preference for the flowers that correspond to the length of their proboscis. Okay. Species that live in the same area tend to have proboscises of different lengths. Okay. What was the same area called when it comes to evolution? Sympatric speciation, right? Sympatric speciation for living in the same area. Okay, so it's not allopatric. Are they helping each other? That wasn't mentioned. It's probably B. They have learned to occupy different type of flowers because, of the different thingy. Okay? Good? All right. Any questions?
No.
So look, guys. If it's morning, it is 1 hour of light. If it's afternoon, it's 8 hours of light. So afternoon has more light intensity. Now, I want you to look at the slope for the mRNA H morning and mRNA H afternoon. Is the slope the same? Are they overlapping slope-wise?
Yes.
They are. So if they're overlapping slope-wise, that means that there's no difference between morning and afternoon. But let's now look at mRNA G afternoon versus mRNA G morning. Which one has a steeper slope? The mRNA G afternoon. So here, the slope is steeper. That means afternoon does affect degradation of mRNA G compared to the morning. But that's not the case for mRNA H. That means a longer exposure of light increases the degradation of mRNA G, but not H, because in H the slopes are the same, but in G the slopes are different. So the answer is going to be choice C. And right now we are studying the length of light exposure. All right. Which of the following observations would provide the strongest evidence that the two plants belong to different biological species? Can they have kids? They could, despite being different species, but the kids can't have kids. Which means what? C. Okay?
They can have kids, but kids can't have kids.
The kids can't have kids, so the kids are sterile.
But why?
That's because they are two different species.
So it's like mules.
Like mules. Mules are the product of a donkey and a horse, but they can't have kids of their own. Make sense? Okay. Long one. Meline, in your own words, can you explain this figure?
Me?
Yeah.
No, thank you.
This is my first time looking at it.
Instead of they can't answer, I can't too.
Your mother was gonna ask me for a report. What do I tell her?
Who's mom? Oh,
What do I tell her? Last time she was happy with the report.
she was very, very happy.
I was happy, she was happy, you were happy.
Everyone was happy. Insulin.
Look. Whoa. That's a long question.
Mister, the thing didn't benefit.
Really?
Yeah. I don't know.
Okay. So, hormones are what? Chemicals. They travel through the bloodstream to signal cells in another part of the body, like insulin is, which induces the uptake of glucose molecules from the blood into the cells. And this way, insulin lowers the overall blood glucose of the body. We have two types of cells, of the bone cells, which can regulate blood glucose levels. Now, binding of the insulin to the insulin receptor will activate a signaling pathway that results in the cells releasing a molecule called OPG, which will then bind to neighboring osteoclasts. In response, the osteoclasts release protons, and this creates a lower pH outside the cell, which then activates a different protein made from the same cell, which is the osteoblast. We have a gene called the ESP gene. As you guys can see, every time there is a gene, they will italicize it, right? Like tilt it. Encodes a protein that alters the structure of the insulin receptor, interfering with the binding of the insulin to the receptor. A researcher created a group of osteoblasts with an ESP mutation, which prevented the production of a functional product. That means the receptor was not harmed, correct? The receptor was not harmed. The researcher then exposed the mutant strain and normal strain that expresses ESP to glucose and compared the level. Well, the normal strain should not let glucose be taken up because the receptor is going to be not active, but the mutant one cannot inactivate the receptor still works. 26. Which of the following best describes the effect of insulin binding to the receptor on the osteoblast cells? Well, what's happening? OPG is made, correct? OPG is made, which then results in more insulin. So it's like a positive feedback mechanism, correct? Okay. Which of the following best explains or best justifies the claim that osteocalcin is a hormone? Now guys, go back to the figure. Osteocalcin, when active, will bind to the osteocalcin receptor, which will then cause the production of insulin. So that's why A is wrong, because osteocalcin does not bind to the insulin receptor, and instead it binds to the osteocalcin receptor. Osteocalcin receptor instead. That's why A is what? Wrong. And a hormone has to result in the cell changing in activity, because a hormone is going to cause a response. Okay? A hormone does what? Causes a response. For example, if it's insulin, it causes the uptake of glucose. If it is osteocalcin, it's going to result in more insulin. Okay? So a response has to happen, and the answer is going to be choice B. The following claims is the most consistent with data shown in figure 2. Okay, so let's look at figure to analyze a bit. Look, the normal strain has ESP, mutant one, no ESP. If we have ESP, insulin receptor is disrupted at some point. If there is no ESP, insulin receptor not disrupted. To disrupt insulin receptor, is that a good thing or a bad thing?
Good.
It's a good thing. Why? Because if you have the ESP gene, it will interfere with the binding of the insulin to the receptor. Maybe at some point you should stop insulin from binding at some point. So in the case of the normal, insulin becomes low, but in the case of the mutant, insulin remains high. Despite the glucose being injected, 20 minutes ago or 40 minutes ago or 60 minutes ago. So why is the insulin still high after the injection? Because look at the pathway. It shot up, right? It shot up, it went down, and then it started to increase. This is wrong. This is wrong, okay? That's not supposed to happen. So actually having ESP is a good thing because you want to keep insulin levels low. And if you guys look at the normal, the insulin levels are kept low at some point. So it's going to be choice A. Question 28. Which of the following was a positive control? A positive control means you already know what the result will be. The researchers already know what the results of normal strains will be, right? But they don't know what the result is going to be of the mutant strain. Always normal strain or no treatment is your control. So it's going to be choice D. Choice D. Okay? A researcher observed that mice from the mutant strain experience low blood sugar. The mutant strain, don't forget, have no ESP. Okay. Which of the following describes the feedback mechanism in the pathway causing the low blood glucose? There's a lot of insulin, which is why the glucose level is low, right? Lots of insulin. So what's the answer? A. Is it A? Can we check? Let's check if it's A. You have a lot of insulin. Okay, good. Confirmed. It's A. Lots of insulin results in low blood glucose because here they say that insulin is a hormone that causes the uptake of glucose molecules. Okay? Okay. A researcher hypothesizes that in mice, two autosomal dominant traits, Q and R, are determined by separate genes on the same chromosome. This reminds me of what? Of?
Linked genes?
Linked genes, exactly. Which basically disobey the law of independent assortment. The researcher crosses mice that are heterozygous for both traits. So I'll just say capital Q, small q, capital R, small r, with capital Q, small q, capital R, small r. What do we get? 9, 3, 3, 1. Now you calculate the expected number of the mice with the combination. How many different phenotypes do we have? 4 different phenotypes, correct? This one expresses what? Dominant. Dominant. This one, dominant.
Recessive.
Recessive. This one,
Recessive dominant.
and this one, Recessive? Recessive. What they want is both dominant traits, which means 9 out of 16. 9 out of 16. But we have 64 offspring. That's 9 times 4, which is 36. Oops. Researchers studying new viruses analyzed the genetic material found in 4 different virus samples to determine the percent nitrogen-based composition of each virus, with data shown. To have a double-stranded RNA, you must have A equaling U, you must have C equaling G. And which one of them is A equaling U and C equaling G? In which one? In which sample?
A equals what?
A is equal to U
C equals to G.
and C
First one, sample 1.
is equal to G. So it has to be sample 1. Not too hard, right?
Yeah.
Goats and sheep. In Kurdish, goat means bizin and sheep means mar. Okay? Cow means chel.
Okay, I didn't know that.
Yeah. Yeah.
Isn't cow manka?
No, that's jamus. That's the bull.
Okay.
But what's horse means asp? Asp?
S.
Asp. See, guys? Know some things.
Mashallah.
Okay. Helka means eggs.
Yeah. Mister, do you think I'm Kurd or Arab?
Look, your mother speaks perfect Arabic, but you never speak it.
'Cause I don't
So
know
I'm
Arabic.
actually confused.
My Arabic teacher says your name is Arabic and you don't even know Arabic.
Farah has an Arabic name.
Yeah.
Yeah.
But I don't know Arabic at all.
So you're not Arab?
No, we're Kurd. But
What your
my
name?
mom, my parents, they grew up in Baghdad,
Ah.
so they know perfect Arabic.
No, the whole world knows your history. The whole world listening to this.
Oh,
The whole world. Okay. Goats and sheep belong to the same family, but different genera. They live together in the same pastures, but the hybrids produced rarely survive. You know what this is called? That's called hybrid breakdown, is what we call it. When such a hybrid does survive, it's usually sterile. That's called hybrid something. Still bad, okay? Which of the following best explains the mechanism that maintains reproductive isolation between the goats and the sheep? Is gene flow prevented? No, because they already mated, so it's not A. They already mated, so it's not habitat isolation. They already mated, so it's not C. So D. Ethylene gas is an organic molecule that serves many cell signaling roles in flowering plants. A positive feedback means producing more of something, right? So, cells of ripening fruit produces ethylene, which activates ripening in other fruits. Maybe. It's not B. Nothing of positive feedback. I don't see any that in C.
it's not D, so it's A.
Yeah. So the ripening
Sounds easy.
activates more ripening. Yeah. Ripening activates more ripening. Okay? It's like watching a reel makes you watch more reels. That's
Why
called
not
positive
TikTok?
feedback.
Yeah, why not? I used to.
TikTok, are they called reels? Or that's only for Instagram? It's called TikTok videos? Just called TikTok, no videos, nothing?
Mister, why don't old people watch TikTok?
I don't.
Yeah, are you calling him old?
No, like,
I don't use TikTok.
Mister, she called you old.
Look, TikTok to me is a waste of time, but to you it's not. Different generations, right?
it's a distraction.
Mister, reels
Big
are
distraction.
really funny though. Reels are funny,
Reels also distraction.
but I rarely watch them.
The
Rarely
fun in
watch them?
general.
Yeah, I mostly watch TikTok.
Alright. A student is investigating photosynthesis in plants. The student planted grass seeds in a tray with 3 sections. and grew the grass under light for 14 days. After 14 days, the student collected all of the grass from section 1 and recorded the mass. Then the student placed a clear cover over section 2. Clear means lets the light through, correct? It can still have photosynthesis. But here, will there be photosynthesis? No photosynthesis. So the mass of the plant decreases. The student then placed the tray back under the artificial lights for 7 additional days. Whoa. Okay. What do we see in here? The mass increased, right? But then the mass decreased. Okay. Based on the data, which of the following is the rate? Rate means what? The slope. From day 14 to day 21. So we're going to say rate. Always put time in the bottom. In day 21, how much was the mass in section 2?
4.2.
9.6, section 2. And how much was the mass in day 14?
5.
We don't have it. We only have for section 1, right? But section 1 and section 2 have the same exact conditions, right? So we're fine. At 14 days, they both are exposed to light. Use the same mass. So minus 5.1, that's going to be 4.5 over 7, which, I don't see that.
The answer.
Oh, wait. I know what happened. I'm finding it per day. Look at the thingy.
Oh, yeah,
it's per week.
45.
It's 1 week.
So we just mine.
7 days, 4 week, right? I mean 1 week, right? So 4.5. So B. Easy. Which of the following best explains the observed difference in mass between the grass in section 1 after 14 days and the grass in section 3 after 21 days? The mass of section 3 dropped, correct? Because the plant was not doing that much photosynthesis. So what do you guys think? Did the grass in section 3 do more photosynthesis? Relax.
No.
Less. D, right?
Yes.
It could not replace what it lost to cellular respiration. Which of the following best explains the connection between energy growth and the maintenance of the ordered system in the experiment? Energy from the light is required. That's correct. The grass obtains the energy for growth from nutrients. Maybe, but light is required for photosynthesis.
Unless it's A.
It's not C.
It's not B. And it's not D.
Why do you say it's A and not B? Because light is important, right? For a plant. And light's not mentioned in D, in B. So it's not B. It's going to be A. Shall we trust the process or shall we trust you?
Me.
Trust her. Come on.
You are the process. 1 second. I have to report, right? 37. Based on the data, which of the following is the most likely effect of a period of increased volcanic activity decreasing sunlight? You're going to have less producers, right?
Yeah.
So it's going to be A. Which of the following is an appropriate null hypothesis? Null means saying the opposite of what you're experimenting.
D.
Yes, that there is no effect. No effect. Okay? No effect is always going to be the null hypothesis. The amino acid in figure 1 is found in a region of a polypeptide that falls away from the water. Ooh, you know what that means? Hydrophobic. And this R group is hydrophobic. Which part of the amino acid most likely contributes to the hydrophobic behavior? The R group.
What? Oh, never mind. Ethylene.
Because look, each amino acid has this one, and has this one, and has this one. What's different is the R group. A researcher observes that when two heterozygous plants with red flowers are crossed, the resulting offspring include plants with red, white, or pink. This reminds me of incomplete dominance. The researcher proposes the null hypothesis that flower color is the result of independent assortment. A chi-square of 7.3. 2 degrees of the freedom. Okay, let's pull up our chi-square. What do you guys think? Is that big enough? Okay. So 2 degrees of the freedom would be here. P-value of 0.05 would be here. That means our critical value is 5.99. But our chi-square value, which is 7.3, is more than 5.99.
So the critical value is less than calculated.
So we're going to reject the null hypothesis.
Yeah.
If your chi-square is larger, you must reject null hypothesis.
A.
Yes. Our critical value is too low, basically. Yeah, and all these texts, I really don't get it. I just posted about the blueprint and everyone's going crazy. I guess they're starting to wake up. A researcher is investigating the effects of a chemical that makes the thylakoid membrane permeable to hydrogen ions. Okay. You know what that means? Bad. Because the membrane has to limit their movement and let them move only at certain times to use the energy to get the ATP. So this is not good at all. This is not good. So what's the answer?
Should be.
Exactly. The membrane should never be allowing all the H plus to move at all times, only at certain times. So this is bad. Ooh, nice one. Researchers analyzed the amino acid sequence of the protein cytochrome C in various groups of organisms and determined the number of amino acid substitutions that have occurred in different groups of organisms. Okay. Based on the data, which of the following organisms are most distantly related? That means they have diverged from each other a long time ago. Birds and mammals. Birds and mammals, they diverged from each other at the year 270 maybe? 270 million years ago. Mammals and reptiles, they diverged from each other, let's say 280 million years ago. Fish and land vertebrates, they diverged from each other maybe like 390 million years ago.
Mister, isn't that on 400?
Yeah, about the same. Insects and vertebrates, they diverged from each other. Oh, that's like very far, like 580 or something.
Yeah, it's that.
So it's that one. 43. Which of the following best explains how continuity of genetic information in cells is ensured across generations? It's all about the DNA. Always. Because DNA has what? 2 strands. So what's the answer?
A.
In the spring and the summer, the fur of the Arctic fox contains a pigment called melanin that gives the fox's fur a dark color. In the fall and the winter, the fur is becoming white. That means the phenotype is dependent on the environment. So what's the answer?
A.
A.
Exactly correct. Here we have what we call the sleep-wake cycle, which I think most of you don't follow. You guys should be in bed like really in bed when it's very black.
So by like 12.
So like by 10 you should be in bed.
Oh.
Yeah, impossible.
No way.
11 you should be like deep sleep, like dreaming and snoring.
Yes, of course.
And 12 like you're just dead. You must be in bed by 10 p.m. And also you must wake up at 6 a.m. Maximum 7:30, even on a weekend. Even on a weekend. And also a holiday like Wednesday.
Mister, after this we have to go study for our chemistry test.
Oh, yeah.
Well, you still have time.
Math.
Look, you must start to like brush your teeth and everything by 9.
Okay, so now I can go to Miss Fatima and tell her Mr. Wasim said that I have to be asleep by 10 p.m. so I couldn't
So I couldn't study.
But you should wake up at 6 a.m. Look, if you sleep at 2.
I'll wake at 4.
Oh, that's why I always suffer when I sleep at 2. Because you must be already asleep by then. If you sleep late, you guys wake up tired, don't you guys think? Always happens to me.
No, I think it's the opposite.
I'm more tired on the weekends because I sleep late and I wake up like late. I don't know. No? Different for you guys?
Yes, that's true.
The less you sleep, I feel like the more energy you have.
Interesting.
You're unique.
I need these genes,
No,
man.
it's actually like that. The more I sleep, the more like tired I am.
Yeah. Alright, the human sleep-wake cycle is regulated by melatonin. They actually sell this in the pharmacy, by the way.
It's all lies. They don't work.
They don't work, right? I tried it. It just gives me headaches.
Exactly.
And like, it makes you sleep in a weird way. You have weird dreams and stuff. Anyways, the synthesis of the melatonin is regulated by light exposure, right? Okay. Ooh. The sleep cycle does not respond quickly to change. Long-term exposure to long periods of light after sunset is going to affect a person. Well, melatonin makes you sleep. So if there is light, you're not going to sleep, correct? You cannot sleep at 10 a.m., can you? There's already a lot of light outside.
We close the curtain.
That's also true. Melatonin synthesis will actually be increased. No. It increased at nighttime. Okay, because here it says it starts to be secreted at nighttime, at 9 p.m., which is like very soon, right?
It's a little inefficient.
It's very, very soon. Oh my god, all these Instagram messages. Okay.
Popular.
No, it's because of the blueprint are like freaking out right now. They're like, what's this?
Block them.
I need customers and followers. Melatonin synthesis will be increased and the person's gonna fall asleep at daytime?
No.
No.
See.
So C. You can't sleep at 10 a.m.
I can. يعني biologically.
Okay. A typical bag of fertilizer contains high levels of nitrogen, phosphorus, and potassium, but trace amounts of magnesium and calcium. Which of the following best matches the fertilizer component with the molecule in which it will be incorporated by the organism in the area? Well, look, nitrogen is important for what? Amino acids and nucleic acids, correct? Based on data in figure 1, which of the following most likely represents the change in energy that occurs when ATP hydrolysis is coupled with the phosphorylation of a substrate? Guys, ATP hydrolysis gives you energy, correct? That means what? It has to be exothermic. So 1 or 2 is the ATP hydrolysis. So it's not 4. And it's not 3. Correct? And the energy released. Are you okay? Yeah.
Me?
Yeah?
Yeah, I'm fine.
You're like shaking. D'allergie.
D'allergie.
That is,
Where? Le d'allergie.
I don't know.
Okay. Is more than energy absorbed. So, are you going to choose 1 and 3, or 1 and 4, or 2 and 3, or 2 and 4? Can 1 and 3 work? No, because the exo has to be more than the endo. Exo must be higher than the endo. So 1 and 3 don't work.
Can
1 and 4 work, 2 and 4 work.
1 and 4.
Yes, there's no 2 and 4, so we choose that. Oof, long one. The brown anole is a species of lizard native to Cuba, which has been introduced into the southeastern US. The range of brown anoles in the US has been expanding, and now they compete with native green anoles. Nice. Okay, I guess they differ in body length. No, actually, they have almost similar body length, but they have different limb length. They have different toe pad size. They also have different cold tolerance, correct? Like the green ones can survive in a colder temperature than the brown ones. Where is the outer group here?
The what group?
Outer group?
The last one.
This one is the outer group.
Yes.
It's the least related to the rest. Based on data in Table 1, which of the following best explains the observed differences in cold tolerance between the brown anoles and the green anoles in the US? Well, the green ones can tolerate a colder temperature. So it's not A.
C. Isn't it? Yeah, C.
In anoles, small toe pads are better for gripping irregular surfaces like the forest floor. So the small one is for the forest floor, large one is actually for the leaves. Let's go back and label that. So the large one is for the leaves, the small one is for the forest floor.
A.
How come it's not the rest? Yeah, look, how can they evolve to have the same toe pad? That's going to increase competition, not decrease it. So it's not that. They will chase away.
No.
They live on different areas. So it is A. Trust you with that. I won't even check. Which of the following is the outer group in the cladogram? We just did it. The one by itself.
Only this one's easy.
I agree.
2024 was like that.
I agree.
Mr.
Wait.
2024.
Oh, the FRQ? Or
this
the
one
full
is
test?
Q. What?
The real test
Oh,
that you
this-
guys took?
Yeah.
Yes.
It was hard.
Yeah.
It was
Mr.
hard.
I was trying to cheat as well, but I couldn't.
Oh.
I was sitting in front of the tray. Oops.
Oh,
Mr. I keep forgetting you.
I
No-
record.
No one watches it.
I'm the only one watching it.
She watches Dreams Team, the one with.
lots of
Mr.
gossip.
Because I'm just joking, gossip.
It's a lot of gossip, yeah. Okay, based on figure 1, which of the following would provide the strongest evidence to support a claim that A. valenciani and A. linotopus? What? A. valenciani? Where's that?
The last.
Are the closest to A. sagre, right? Well, the best evidence to look at DNA sequence or amino acid sequence. Never look at morphology. Never. Based on the cladogram in figure 1, which of the following conclusions about the evolution of the anole species is the most reasonable one? Did they undergo convergent evolution? No, they had different features, right? They had different features. So what's the answer going to be?
B.
Exactly correct. Beta thalassemia is a disorder caused by mutation in the HBB gene. The protein is missing many amino acids at the carboxyl terminal. How could you lose a carboxyl terminal? What do you guys think? Oh, a stop codon before its time. Correct? A stop codon before its time. A common test for liver function involves sprinkling sulfur powder onto a sample of urine. Sulfur powder sprinkled on a sample from an individual with impaired liver function will sink because the urine contains a high level of bile salt. But the same powder or normal urine is going to float. Why bile salts cause sulfur powder to sink? It sinks, right? It sinks because it doesn't have any surface tension, correct?
Yeah. So it's A.
So it's A. Shall we check that one?
Mister, it's correct.
I felt like it was first.
Have you guys memorized the answers, or how do you guys do it? What's your secret?
Mister,
We've been studying since September.
Shall we continue the rest for next time?
We have, it's until 60?
Yeah.
What, 55?
I think you guys, yeah. This will take us sometime.
Mister, just say you don't want to do the whole thing.
Balakis, I'm not fasting. For me, it's easier. Barojnabin.
Well,
See, guys, I know Kurdish.
Mister, it's how much? 55 or 60?
55. Type is a representation of all metaphase chromosomes in a sample of cells from particular individual. Is this person a male or a female?
Male.
Male. How do you know?
X and Y.
They have X and Y. Do they have 46 chromosomes or 47?
47.
Yep. They have 3 in the chromosome 21. This person has what we call a Down syndrome because they have trisomy 21. Which of the following most likely explains how the chromosome circled in figure 1 could cause a genetic disorder? Well, the person has an extra chromosome, correct? Okay. The extra chromosomes. Wait, can the extra chromosome cause other chromosomes to become triploid?
No.
No. Okay. Can the extra chromosome interfere with the pairing of the homologous chromosomes?
Yes.
Probably could, right? Because homologous means 2. That's going to be 3.
I think the answer is B.
Are we wrong?
I think so. Let me check.
Let's check. Oh shoot, the answer is wrong. I mean, look. Why is the answer B? If you have an extra chromosome, that means you're going to have 3 instead of 2. That means you're going to have more RNA from certain genes. And that's just not normal. You must have equal amount of RNA. But now you're going to get more RNA in certain genes because of this extra chromosome. Okay? Very good. And yeah. Okay. Oh, by the way, why is C wrong? Because they talked about homologous chromosomes, which is only during meiosis. But how about mitosis? We don't discuss them, so don't pick C. Which of the following is the strongest evidence supporting the endosymbiont hypothesis? Which is the hypothesis that the mitochondria and the chloroplast were at some point acquired by a bigger prokaryote and they became a permanent part of it. And over time, this prokaryote became a eukaryote. They have their own DNA, and they also divide independently of the cell. Also, they have a double membrane. They also have their ribosomes, which happen to have a smaller size than a eukaryote's ribosomes. Time for face washing? She never accepts. Which of the following best explains how the expression of a eukaryotic gene encoding a protein will differ if the gene is expressed in a prokaryote? Guys, a eukaryotic gene has introns and exons. But a prokaryotic gene only has exons, no introns. So, could a prokaryotic cell be able to remove the introns? No, because it was never taught to remove them. A prokaryote only has exons. So, the prokaryotes simply cannot remove these introns. Okay? Good, 1 second. All right. Low concentration of cadmium in the water negatively affects steelhead trout. In a river community, steelhead trout are part of the trophic structure shown in figure 1. Which of the following is the most likely immediate effect of an increase in runoff containing cadmium? Well, if you have more cadmium, you should have less steelhead trout, correct? Of course, cadmium is going to help the green algae. It's going to help it. So it's not going to be A, because algae live on runoff. Okay. Now here it says that if you have small concentrations of cadmiums, you expect to have a negative effect on steelhead trout. Imagine if you have a large amount of cadmium. You can have a huge decline in the trouts, a huge decline, right? Why? Because the algae are going to overgrow and they're going to die at some point and they will be decomposed by decomposers who will use up all the O2 in that river, killing the trout. Poaching is illegal. It means hunting or capturing wild animals. It's illegal. We're talking about tusks here of an elephant, and the size of the tusk is heritable. So look, if the poachers are targeting elephants with a large tusk, over time the ones with the large tusk will be hunted and killed, and the ones that survive are the ones that were not hunted because they had a small tusk. Correct? So you must not have more tusk size.
So either B or D.
You must have a smaller tusk size. So the answer is B. A scientist adds a chemical to a culture of dividing cells in order to disrupt DNA replication. The replicated DNA produced by the cells is double-stranded, but some sections like covalent bonds. They like what we call the phosphodiester linkage. Okay. So isn't DNA ligase responsible for joining
Yes.
the covalent bond? But right now they're not being joined. That means DNA ligase is not working because it's DNA ligase's job to do that.